Hello everyone, my name is Alexa Mogus. I am a current grad student at Grand Canyon University in the Chem 530 Concepts of Biochemistry class. In this presentation today, we are going to discuss cell cycle regulation. Cell cycle regulation is significant for proper cellular homeostasis. It is necessary so that cells may constantly undergo cell division. This is achieved by the activation and inactivation of series of proteins that control the replication and segregation of DNA equally to daughter cells. Okay, so let's take a look at our objectives for this presentation. So first off, we are going to review the cell cycle. We are going to dive into each of the four phases of the cycle, the phases being gap 1, synthesis, gap 2, and mitosis. And also we are going to talk about the importance of the gap 0 phase. Next, we are going to discuss the checkpoint control system. So specifically, we will discuss the three major checkpoints throughout the cell cycle, and this includes the G1S checkpoint, the G2M checkpoint, and the spindle checkpoint. Next, we will talk about cell cycle signals. So we will talk about cyclins and cyclin-dependent kinases and their known roles in the cell cycle. And lastly, we are going to talk about cell cycle regulation. So first, we are going to talk about positive regulators, and this includes our cyclins and our cyclin-dependent kinases. And then we are going to talk about negative regulators. So this will include retinoblastoma, tumor suppressors, and tumor protein P53. Let's take a look at gap 1, or the G1 phase. So the cell cycle, it is ordered in a series of events involving cell growth and cell division that produces two new daughter cells. So a cell moves through a series of phases each time it divides. So let's take a look at the image here on the screen. In the center, we have our interphase period, and this is a period of growth that occurs between cell divisions. And the three stages of our interphase are G1, S, and G2. But we're going to talk about G1 right now. So the G1 phase is the first stage of interphase, or the first gap where the cell is rapidly growing. As the cell is growing, it produces proteins and replicates organelles. And the G1 phase is the longest phase of the cell cycle. The cell cycle is accumulating the building blocks of chromosomal DNA and associated proteins, as well as collecting enough energy to prepare for replication of each chromosome. Important events such as transcription of the three types of RNAs, synthesis of regulatory proteins, enzymes required for DNA synthesis take place during this phase. Okay. So that occurs here in our G1 on the screen, and this is where, just to recap, our cellular contents, excluding the chromosomes, are duplicated. And here you can see that we start off with our first two little chromosomes. So let's bump over to the G0 phase, and this is an alternative phase in which cells are not actively preparing to divide. The cell is in a quiescent or inactive stage. Most mature human cells enter the G0 phase. For example, liver cells can enter the G0 phase temporarily until an external signal triggers the onset of G1. Nerve and muscle cells are highly specialized that never divide, and they remain arrested in the G0 phase. At this point in time, the cell has two choices: to divide or not to divide. Between G1 and S phase, the cell decides if it wants to grow. The cells that do not divide do not go through the S phase or G2. They stop at G1 or G0. In this next slide here, we are going to talk about synthesis, or the S phase. So during S phase, DNA replication results in the formation of two identical copies of each chromosome, or also known as sister chromatids. And these sister chromatids are attached at the centromere. Each chromosome is made up of two sister chromatids in a duplicated chromosome. There is synthesis of proteins required for DNA packaging, such as histones, and the centromere is duplicated during the S phase. Therefore, two centromeres will give rise to the mitotic spindle, which is the apparatus that orchestrates the movement of chromosomes during mitosis. The centrosome consists of a pair of rod-like centrioles, and they help to organize cell division. By the end of S phase, each chromosome has two DNA molecules and a duplicate set of genes, and the S phase lasts for about between 6 and 10 hours. So just to recap on our screen here, our little figure, so for the S phase, each of the 46 chromosomes is duplicated by the cell. As you can see on the left here, each chromosome is separated and duplicated and becoming sister chromatids. And then the image here on the bottom left just shows our homologous chromosomes separately, and then going through DNA replication, they become sister chromatids. In our next slide here, we are going to talk about gap2, or the G2 phase. So in the G2 phase, or second gap, the cell replenishes its energy stores and synthesizes proteins necessary for chromosome manipulation. This phase is basically where the cell prepares for division. At this point, the cell increases in size and contains double the DNA. Some cell organelles are duplicated, and the cytoskeleton is dismantled to provide resources for the mitotic spindle. The G2 phase ensures that any errors in the chromosomes are repaired and that the cell is ready to proceed before moving on to the next phase in the cycle. So again, on our figure here, if we're going to look at G2 on the top, a recap, the cell double checks the duplicated chromosomes for error, making any needed repairs before it enters mitosis. On this slide here, we are going to talk about the M phase, or mitosis. So if you take a look at the slide here, we're going to look at our figure that we've been looking at, and we can see on the right, the top right, we're going to enter mitosis, and then we're also going to talk about cytokinesis. And the overview of each phase of mitosis can be seen on the image, and if you want to follow along with me, I will explain what happens during each section of this mitosis. So the multi-step mitotic phase is where the cell nucleus divides and the cell components split into two identical daughter cells. Duplicated chromosomes are aligned, separated, and moved to opposite poles of the cell. The M phase is the shortest phase of the cell cycle, and it takes about 1 hour for completion. The first portion of the M phase is mitosis, composed of 5 stages, which accomplish nuclear division. So let's start off at prophase. So our chromosomes begin to condense and become visible. The spindle fibers will emerge from the centrosomes, and the nuclear envelope will begin to break down. And then our centromeres will start moving towards opposite poles of the cell. Next, entering pro-metaphase, our chromosomes will continue to condense at this stage, and our kinetochores will begin to appear at the centromeres. The mitotic spindle microtubules attach to the kinetochores during prometaphase. Our next phase we are going to enter is metaphase. So at this phase, the chromosomes are lined up at the metaphase plate, and each sister chromatid is attached to a spindle fiber originating from opposite poles. Then we will reach anaphase, And this is where our centromeres will split into two. Our sister chromatids, or our chromosomes, are pulled towards the opposite poles of the cell. And certain spindle fibers will begin to start elongating the cell. The last stage of mitosis is telophase. At this point, the chromosomes will arrive at opposite poles and begin to condense. They will begin to decondense. The nuclear envelope material will begin to surround each set of chromosomes. The mitotic spindle will begin to break down, and the spindle fibers will continue to push the poles apart. So the second portion of the mitotic phase is cytokinesis. And cytokinesis is the physical separation of the cytoplasmic components into two daughter cells. So in animal cells, for cytokinesis, a cleavage furrow will separate the two daughter cells into two. In this slide here, we are going to talk about cell cycle control. So the cell cycle is controlled by stop and go chemical signals at three critical points. This is the G1, G2, and M phase points, and we're going to discuss those more in detail in the next slide. So these signals indicate if key cellular processes have been completed correctly or need to be stopped until conditions are favorable. The essential processes of the cell cycle, such as DNA replication, mitosis, and cytokinesis, are triggered by the cell cycle control system. So, using the analogy of a washing machine, the cell cycle control system is shown as a central arm, or the controller. And we can see this here on the image that I have provided on the right. So this controller rotates clockwise, triggering essential processes when it reaches specific points on the outer dial. The control system of most cell cycles and sophisticated washing machines is responsive to information received back from the processes it is controlling. So sensors, for example, detect the completion of DNA synthesis or the successful filling of the wash tub. If some type of malfunction prevents the successful completion of this process, signals will be sent to the control system to delay the progression to the next phase. And this delay will provide time for machinery to be repaired and prevent disaster that may result if the cell cycle progressed prematurely to the next stage. In this next slide here, we are going to talk more in depth about the three major checkpoints of the cell. So the cell cycle is controlled at three different checkpoints. These checkpoints occur near the end of G1, at the G2M transition, and during metaphase. So let's talk about the G1 checkpoint first. So this checkpoint determines whether all conditions are favorable for cell division to proceed. The G1 checkpoint is also known as the restriction point, and it is the point at which the cycle irreversibly commits to the cell division process. The integrity of DNA is assessed at the G1 checkpoint, and if a cell does not meet all requirements, it will not be released into the S phase. Next, let's dive into the G2 checkpoint. So this checkpoint bars the entry to the mitotic phase if certain conditions are not met, and proper chromosome duplication is assessed during this checkpoint to ensure replicated DNA is not damaged. Finally, our third major checkpoint is the M checkpoint, and this occurs near the end of the metaphase stage of mitosis. It is also known as the spindle checkpoint because it determines if all the sister chromatids are correctly attached to the spindle microtubules. Because the separation of sister chromatids during anaphase is an irreversible step, the cycle will not proceed until the kinetochores of each pair of sister chromatids are firmly anchored to the spindle fibers arising from the opposite poles of the cell. And then as we can see here on the slide, I do have an overview image of our control system and how it moves clockwise, and then it shows the points in which each checkpoint correlates with the different phases of the cell cycle. Alrighty, so our next 3 slides that we're going to talk about in this presentation are all going to tie into one another. But first off, in this slide, we are going to focus on cyclins. So cyclins are a family of regulatory proteins that control the progression of the cell cycle. So in order to drive the cell cycle forward, a cyclin must activate or inactivate target proteins inside of the cell. Cyclins drive the events of the cell cycle by partnering with a family of enzymes called cyclin-dependent kinases, and we'll discuss more detail about those in the next slide. So if we take a look at our graph here, we have variations in the activities of specific cyclin-dependent kinases during the cell cycle, and those can be observed here. So on our graph, it goes by the x-axis is going to represent time, and then our y-axis is going to represent our kinase activity. Okay, so let's talk about the blue line here. So cyclin E, cyclin-dependent kinase 2 activity peaks near the G1 phase, S phase boundary, when the active enzyme triggers synthesis of enzymes required for DNA synthesis. Cyclin A, cyclin-dependent kinase 2 activity rises during the S and G2 phases, and that can be seen here in the green line. So once that rises during those S and G2 phases, it will then drop sharply into the M phase as cyclin B, cyclin-dependent kinase 1 peaks. And you can see that here on the red image on the screen. So cyclin D is not shown in this graph, but it is active throughout the cycle as long as the growth factor is present. Increases in the concentration of cyclin proteins are triggered by both external and internal signals. Once the cell moves on to the next stage of the cycle, the cyclins that were active in the previous stage will become degraded by cytoplasmic enzymes. In this slide, we are going to focus on cyclin-dependent protein kinases. So, of the many proteins involved in the cell cycle control, cyclin-dependent protein kinases are among the most important. Cyclin-dependent protein kinases are a family of multifunctional enzymes that can modify various protein substrates involved in cell cycle progression. Specifically, the cyclin-dependent protein kinases are enzymes that phosphorylate or attach phosphate groups from ATP to specific target proteins. The attached phosphate group acts like a switch, making the target protein more or less active. There are different types of eukaryotic cells that contain different types and numbers of cyclin-dependent protein kinases. For example, yeast can only have a single cyclin-dependent protein kinase, whereas vertebrates have 4 different ones. Cyclins activate our cyclin-dependent kinases, which control cell cycle processes through phosphorylation. In the absence of the cyclin, the catalytic subunit is inactive. Cyclin-dependent kinases must also be in a particular phosphorylation state, with some sites phosphorylated and others dephosphorylated in order for activation to occur. So when a cyclin attaches to a cyclin-dependent protein kinase, it has two important effects. So it activates the cyclin-dependent protein kinase as a kinase, and it also directs the cyclin-dependent protein kinase to a specific set of target proteins. And these are ones that are appropriate to the cell cycle period controlled by the cyclin. G1 and S cyclins send cyclin-dependent protein kinases to the S phase targets promoting DNA replication, while the M cyclins send cyclin-dependent protein kinases to M phase targets, making the nuclear membrane break down. Cyclin-dependent protein kinase levels remain relatively constant across the cell cycle, but activity and target proteins change as levels of the various cyclins rise and fall. So if we take a look at the images on the slide here, on our left we have an image of a cyclin and a cyclin-dependent protein kinase, and they are inactive at part A. At part B, they are partly inactive, but they are beginning to attach here. And then finally at part C, they are fully active, in which it is activating a phosphate. In the image to the right here, it shows that with no cyclin, the cyclin-dependent protein kinase is inactive, and its targets, specifically targets 1, 2, and 3, are not working because the S phase factors are off. However, if we jump to the image on the right here, we have the G1 and the S cyclins, and in this case, the cyclin-dependent protein kinase is activated, and its targets are phosphorylated with our phosphate group, and therefore the S phase will begin and DNA replication enzymes will become activated. In this next slide, we are going to talk about positive regulation. So positive regulation of the cell cycle is achieved by both cyclins and cyclin-dependent protein kinases. So these proteins are responsible for the progress of the cell through various checkpoints. In order for full activation, the cyclin-dependent protein kinase cyclin complex must be phosphorylated in specific locations. Our cyclin-dependent protein kinases are enzymes that will phosphorylate other proteins by changing its shape. The proteins phosphorylated by the cyclin-dependent protein kinases are involved in advancing the cell to the next phase. The levels of cyclin-dependent kinase proteins are relatively stable throughout the cell cycle. However, the concentrations of cyclin fluctuates and determines when the cyclin-dependent protein kinase cyclin complexes form. There are different cyclins and cyclin-dependent protein kinases that bind at specific points in the cell and regulate different checkpoints. So since the cyclic fluctuations of cyclin levels are based on the timing of the cell cycle and not on specific events, regulation of the cell cycle usually occurs by either the cyclin-dependent protein kinase molecules alone or the cyclin-dependent protein kinase cyclin complexes. So without a specific concentration of fully activated cyclin and the cyclin-dependent protein kinase complexes, the cell cycle cannot proceed through the checkpoints. So at this point, I'm going to read through the diagram shown on the image here. So starting off, our cyclin will begin to bind to our cyclin-dependent protein kinases, and our phosphate donating protein will attach. Therefore, our cyclin-dependent protein kinase complex will become phosphorylated. And then it will become activated, the cyclin-dependent kinase and the cyclin complex will phosphorylate target proteins. So at this point, our activated protein will begin to advance the cell cycle, and it will end up being that target protein and that phosphate donating protein. So although cyclins are the main regulatory molecules that determine the forward momentum of the cell cycle, there are several other mechanisms that fine-tune the progress of the cell with negative rather than positive effects. So these mechanisms essentially block the progression of the cell cycle until problematic conditions are resolved. Molecules that prevent the full activation of cyclin-dependent kinases are called cyclin-dependent protein kinase inhibitors. So many of these inhibitor molecules directly or indirectly monitor a particular cell cycle event. The block placed on cyclin-dependent protein kinases by inhibitor molecules will not be removed until the specific event that the inhibitor monitors is completed. So we will discuss more about the negative regulation of the cell cycle in the next slide. Okay, so in this slide, let's talk about negative regulation. So the second group of cell cycle regulatory molecules are negative regulators. Let's recall that in positive regulation, active molecules such as cyclin-dependent protein kinases and cyclin complexes cause the cell cycle to progress. In negative regulation, active molecules halt the cell cycle. So the best understood negative regulatory molecules are retinoblastoma protein, P53, and P21. All three of these regulatory proteins were discovered to be damaged or non-functional in cells that had begun to replicate uncontrollably or become cancerous. In each case, the main cause of the unchecked progress through the cell cycle was a faulty copy of one of the regulatory proteins. For this reason, the retinoblastoma protein and other proteins that negatively regulate the cell cycle are sometimes called tumor suppressors. So the retinoblastoma protein, P53 and P21 act primarily at the G1 checkpoint, where the cell determines whether it has adequate energy reserves or is large enough, and whether there is damage to the genomic DNA. So the P53 protein has a major impact on the commitment of a cell to divide. If damaged, DNA detected at the G1 checkpoint, P53 will halt the cell cycle and recruit enzymes to repair the DNA. If the DNA cannot be repaired, P53 can trigger apoptosis or cell death to prevent the duplication of damaged chromosomes. As P53 levels rise, the production of P21 is triggered. P21 enforces the halt in the cell cycle dictated by P53 by binding to and inhibiting the activity of the cyclin-dependent protein kinase complexes. If the cyclin-dependent protein kinase complexes are inhibited, the cell cycle cannot move forward. As a cell is exposed to more stress, higher levels of P53 and P21 accumulate, making it less likely that the cell will move into the S phase. So the retinoblastoma protein exerts its regulatory influence on other positive regulator proteins. Retinoblastoma protein monitors cell size. So in the active state, the retinoblastoma protein binds to proteins called transcription factors. Transcription factors turn on transcription of specific genes, allowing for the production of proteins encoded by that gene. So when the retinoblastoma protein is bound to transcription factors, production of proteins necessary for the G1S transition is blocked. As the cell increases in size, the retinoblastoma protein is slowly inactivated. the retinoblastoma protein will release the transcription factors, which can now turn on that gene that produces the transition protein, and this particular block is removed. For the cell to move past each of the checkpoints, all positive regulators must be turned on, and all negative regulators must be turned off. So a giant takeaway of what I just explained here about negative regulation, I want you to know that if the p53 protein detects DNA damage, which can't be repaired, it tells the cell to commit suicide. When p21 is produced in response to p53 and stops the cell cycle by inhibiting cyclin-dependent protein kinases and cyclins. The retinoblastoma protein blocks production of proteins necessary for the cell cycle to proceed. And the images on the screen here just provide an overview of retinoblastoma protein regulation of the cell, and then on the left here we have the normal p53 versus the mutated p53 and what kind of happens during each one. Okay, so finally reaching this very last slide of the presentation, we are going to review everything we talked about. So looking at the top left image here, we are going to review the 4 phases of the cell cycle. And this includes, first off, our gap 1 phase, where cellular contents are replicated. Next is the synthesis phase, and our DNA replication creates our sister chromatids here. The gap 2 phase is the phase that repair of DNA replication occurs. And then finally, we have our mitosis phase, which is the creation of our 2 identical daughter cells. And then off to the side here, we do have our G0 phase, our resting phase, which is where we have cell cycle arrest. Okay, jumping over to the top right image here, we are going to talk about the 3 major checkpoints. So our G1S checkpoint maintains the integrity of DNA. The G2M checkpoint ensures proper chromosome duplication. And then finally, our M checkpoint or spindle checkpoint focuses on the attachment of each kinetochore to a spindle fiber. In the bottom left image, we are going to review cyclins and cyclin-dependent protein kinases. So different cyclins bind to and activate different classes of cyclin protein-dependent kinases. Essentially, the cyclin binds and activates cyclin-dependent protein kinases. So our cyclin-dependent protein kinases activate target proteins to regulate the cell cycle. After the event, the cyclin is degraded, and then our cyclin-dependent protein kinase is rendered inactive. And finally, jumping to the right image on the bottom here, we're going to talk and review about positive and negative regulation. So positive regulation activates cell division when conditions are favorable. Negative regulation inhibits cell division when conditions are unfavorable. Ultimately, proper cell division, is this careful balance between both positive and negative regulation. Okay, and here is my references, and I wanted to thank everybody for watching.